Thank you. Welcome back. Actually, you all look very fresh-faced and ready to go, which is really fantastic. I'd like to welcome everybody back to the second day of this meeting here at the Novotel in Newcastle. So this morning we've got a very exciting day ahead of us with a series of different areas grouped together. So we're going to start off with a morning session on the autonomic nervous system. And before I go and introduce our plenary speaker, I just wanted to let people know that Ed from the Science Media Centre is going to run a workshop over lunchtime on science and the media. So if anybody would like to participate in that workshop, it's going to be in here at 1 o'clock. But could you let Sonia know at the coffee break if you'd like to participate so they've got some idea of numbers? You can go and get lunch and then come back and bring your lunch into here. But 1 o'clock is when it's going to kick off. So let Sonia know if you'd like to participate. Brilliant. So we're going to kick off with a very good friend of mine, Professor Nijs, who has very kindly come over from the Free University in Brussels to speak to us today. Jo and I go back a long way. We've shared the platform at many meetings here and in places around Europe. And I know that Jo is going to give us a great talk today. He's going to talk to us about the autonomic nervous system and the work that he's been doing at the Free University with his group there. He's a physiotherapist by background and heads up the pain motion group there. So with great pleasure, I'd like to introduce you to Jo.
Thank you, Julia. Yesterday when I arrived here in the hotel late in the evening, the door to the poster session was still open, and I noticed only after getting a beer from the bar, and then I was able to take my beer and check out the posters, which was a kind of a private poster session for me, which was really good. So I was really impressed by the quality of the work presented there. So congratulations to those presenting the posters, and also for the organization committee for putting down this wonderful event. It was, all that I can tell, perfectly organized. I've heard some very good feedback from yesterday, so looking forward to today. Oh, this is the wrong presentation. It's the other one.
This is
this is the large size one. So we need the other one because if we use this one, then several slides are messed up by transforming.
This is the one that we saved. Do you want to give me your? I'll just run it from your stick.
Sorry.
Yeah. How is it? It's almost playing. So I saved it as Wednesday morning. That's the one we just had open. But I can run it from the stick if you.
Oh, it's fine. That's it.
Sure.
So, fatigue, sleep disorders, sleep problems, pain, concentration difficulties, those are all characteristic features of patients with chronic fatigue syndrome, but they're not really specific. They're seen in whatever kind of chronic illness, and there's only one rather specific symptom, and that's the really dramatic increase in pain and fatigue following exercise. So we see that in other chronic illnesses also, but not to this extent as we see it in patients with chronic fatigue syndrome. And that's why this is becoming more and more an important and really characteristic, specific feature of this illness. And that's why we have put so much emphasis on this specific symptoms, and also to look at the physiological background of post-exertional malaise, which is the common term for this dramatic increase in symptoms following exercise or whatever kind of vigorous physical activity. So hopefully in the next couple of 40 minutes I will be able to show you that we have actually made progress in understanding of this dramatic increase in symptoms following exercise. And we will address a number of features, of course, including the autonomic nervous system, but also including the possibility of the involvement of dysfunctional cerebral blood flow, the immune system, patient's expectations, and the hyperexcitability of the central nervous system termed central sensitization. Let's kick off with the immune system. We know that even in healthy people, the body of human beings responds to exercise with an immune response, which can be viewed upon as kind of a low-grade inflammatory response. That's a real normal response. And that's why it's important to continue doing exercise, because it gives some fuel to your natural killer cells, for instance. So that's a real normal response to exercise. So in response to acute exercise, so one bout of exercise, you see an acute low-grade pro-inflammatory response, but you have to be aware that exercise training or long-term sports involvement actually decreases this inflammatory response to exercise. Your body adapts to it. Not only in healthy human beings, but also in patients with chronic illnesses, like the ones listed on the slide. But I have to say that this has not been shown to occur also in patients with chronic fatigue syndrome. So we need large studies to actually examine whether the immune response to exercise in the body of patients with chronic fatigue syndrome also adapts to long-term exercise training. What we do know is that there is a body of literature out there studying the acute response to exercise. So the response of the body of patients with chronic fatigue syndrome in terms of immune response to one bout of exercise. And summarizing all these studies in a systematic way resulted in this conclusion that there is actually a enhanced, so a more stronger response of the immune system to exercise in the body of patients with chronic fatigue syndrome. And this more pronounced immune response to exercise is focused on more complement activation and alpha gene expression, and also more oxidative stress in response to exercise. And this correlates to increase in pain, increase in fatigue following exercise. And of course, this is one of the complement factors, but I'm not telling you anything new. You recognize this probably as one of the key complement proteins. Very beautiful one. And it's again on that slide. But it's not so fun if you have too much of it following exercise. So getting back to whether this actually explains at least part of the post-exertional malaise seen in patients with chronic fatigue syndrome. We know that too vigorous exercise produces too much of those proteins. And of course we know that too vigorous exercise also winds up the excitability of the central nervous system. We know that at rest, the central nervous system in patients with chronic fatigue syndrome, like it is in fibromyalgia, is hyperexcitable. But there might be a connection between the two, as suggested in this slide. We don't know for sure, but at least we have data from fundamental research, from animal research, showing that these types of cells in the central nervous system, microglia, are actually able to pick up these complement proteins, and by picking them up, they will further increase the excitability of the central nervous system. But we don't know for sure whether these two separate observations are interconnected in patients with chronic fatigue syndrome. But at least on a theoretical basis, they can be connected to one another. Looking more into those cells, because we now know that the microglia and also the astrocytes are more important than the neurons. Within the central nervous system, we've always given a lot of attention to the neurons, but we now know that these microglia and astrocytes are even more important, because they're the ones sending out the commands towards the neurons. It's like all the different countries in the European Union, And all the separate governments, they're becoming less and less important because, of course, it's the central European government who's saying what each country should do. And I guess you guys are right in kind of opposing to that. So I can slightly understand how you think. But getting back to the nervous system. So we now realize that the microglia and the astrocytes are probably more important than the neurons. And they command the neurons. And one of the commands that they send towards the neurons is sending brain-derived neurotrophic factor. That's one of the main factors, one out of many factors that they produce. And by doing so, normally this is a healthy thing. When there is more brain-derived neurotrophic factor, that's a healthy thing. But we know from a body of literature that's also one of the key factors which is produced in too much quantity. When this is the case, when there's too much of brain-derived neurotrophic factor, that this will actually increase the excitability of the central nervous system, as seen in fibromyalgia, as seen in chronic fatigue syndrome. So, luckily we are able to study this more in detail, and don't mind the name. It's not only produced by the brain, it's also produced by immune cells, also by other peripheral cells, but often these kind of substances are named after the way that they were originally discovered. And it was originally discovered as a neurotrophic factor produced by brain cells. It's like, but you shouldn't always take the name literally, like Julia introduced me as Joe Nijs. I'm not always nice. So. We also know from animal research, but we don't know whether this is actually happening in chronic fatigue syndrome. We should study it more in detail. That exercise therapy normalizes this excessive amounts of brain-derived neurotrophic factor and also the glial hyperactivity resulting in more BDNF. Another thing that these microglia are doing is actually responding to poor sleep. And they do that quite fastly. Even after 1 or 2 nights of poor sleep, they respond by producing a lot of inflammatory substances. So this might explain some of the observations which were done and were published in very nice journals, looking at showing some evidence of low-grade inflammatory responses in the brain of patients with chronic fatigue syndrome. This might be related to the poor sleep that these patients are actually experiencing. So it's great to see that this afternoon there will be a session on sleep, and also that there are some very nice posters in the other room about sleep and also how to manage sleep in patients with chronic fatigue syndrome, because this is a really crucial issue. And when you look at the neurobiology of sleep disorders, so we know that these microglia produce pro-inflammatory substances. Anyone recognizing this one, this pro-inflammatory substance? It's a very common one. It's interleukin type 1, but anyway, also interleukin type 6 and glycolysis factor and nitric oxide is produced by the glia in response to sleep deprivation. And we know that this low-grade inflammatory response in the central nervous system in response to sleep deprivation is linked to the increased excitability of the central nervous system also. So again, these different observations may be connected to one another, explaining more about how patients with chronic fatigue syndrome end up in this terrible illness. So we've talked about the immune system, we've talked about the hyperexcitability of the central nervous system, only 2 pieces of the puzzle in terms of post-exertional malaise in patients with chronic fatigue syndrome. And let's get more into the topic of this session, the autonomic nervous system. This slide is trying to focus on the vagus nerve, which you can see over here, and it's getting to all these internal organs. And the vagus nerve is the most important parasympathetic nerve in our body. It represents 70% of the parasympathetic branch of the autonomic nervous system. And this is a very interesting nerve because it's not only a nerve sending signals from the brain towards the peripheral tissues. including to decrease inflammatory responses, because that's one of the major functions of the vagus nerve and the parasympathetic nervous system in general, but also it picks up signals, including pro-inflammatory substances, from the periphery and informs the brain about any ongoing peripheral inflammation or infection or whatever. So it informs the brain when something is wrong, and it triggers a kind of sickness response with a lot of fatigue and a lot of pain and illness behavior. So we were wondering whether this is functioning properly in response to exercise in patients with chronic fatigue syndrome. We know, as I have explained, that exercise in chronic fatigue syndrome triggers more complement proteins and other inflammatory and other immune factors. And normally we should have a vagus response or a parasympathetic response to exercise also, especially in the recovery phase following exercise. The recovery phase, when you stop exercise, then the parasympathetic nervous system should make your body recover from exercise. And one of the ways it is recovering from the exercise response to whatever kind of physical activity is by shutting down the immune response to exercise. So one important way to look at this is whether patients having poor responses to exercise, whether this mechanism is functioning or not. We don't know, so we examine it. And what we know from looking at the nervous system, the autonomic nervous system in general, and especially the work done by Julia Newton and a group here in Newcastle, is that there is malfunctioning of the autonomic nervous system, including too much sympathetic nervous system activity. And this is one of the conclusions from a systematic literature review. This is one of the conclusions of a MAA study, funded study, showing that there is actually a problem in the parasympathetic recovery following exercise. So the recovery phase following exercise is lacking proper parasympathetic activity. And this is in line with what Julia and Professor Jones has shown years ago, and what Laura and it relates to the recovery phase following exercise. And we know from her work that the recovery phase following exercise is probably the most important part to look at, and also that the lack of intramuscular pH recovery is related to autonomic dysfunction, and even in local exercises, as she has studied back then. So we wondered whether this also relates to the symptom increases following exercise. And we did actually found nice interactions between the reduced activation of the parasympathetic branch of the autonomic nervous system following exercise and the increase in pain following exercise in patients with chronic fatigue syndrome, and also the inability of the brain's capacity to activate top-down pain inhibitory action following exercise. So it is actually interrelated to previous observations done in patients with chronic fatigue syndrome. So we know that this mechanism is not working properly, and again, this is an important piece of the puzzle, explaining the increase in pain and fatigue following exercise and the lack of patients with chronic fatigue syndrome to properly respond to different types of physical activity. Getting back to the title, one thing which has only been briefly mentioned is the part of the title displayed in red, the dysfunctional exercise-induced pain inhibitory action in response to exercise as typically seen in patients with chronic fatigue syndrome. Because, and you might wonder what this is. Well, this slide explains what activation of pain inhibitory mechanism in response to exercise is. This guy is clearly in physical activity, doing exercise, and he clearly has a very horrible injury. He's got a lot of nociception, but he's not feeling any pain. And that's because of several reasons. And I'll get back to this one because you're all thinking of the bull, but I'm thinking right now of the brain of this guy who's activating powerful pain inhibitory action. Why? And that's the reason why, or one of the reasons why he's not feeling any pain. So normally when you do exercise, you should activate several key brain areas, including the periaqueductal gray, but also including the dorsolateral prefrontal cortex and also including the thalamus. All these key centers in the brain activate powerful top-down pain inhibitory action, making sure that you are not feeling a lot of pain during exercise and are able to feel much more pain in response to exercise. And this results in an increase in pain threshold following exercise. And we have studied this repeatedly, and you see the different locations throughout the body where we studied it. And this is the normal response to exercise in healthy human beings. And this is what we see in patients with chronic fatigue syndrome. They do not increase at all, they stay the same, or they further decrease. So this is clear evidence of malfunctioning of pain inhibitory action in response to exercise. You might think, well, this is not really specific to chronic fatigue syndrome. Well, it actually is. When you look at this table, this part of the table, so this column summarizes or provides several medical entities, several medical diagnoses. This table provides you with the response of those people to local strengthening exercise. And when there is a question mark, this hasn't been studied. When there is a plus, it implies that patients with shoulder myalgia, they respond with an increase or activation of pain inhibitory action in response to this local type of exercise. And when there is a minus, it implies that the pain inhibitory action is not activated in response to that type of exercise. Of course, aerobic exercise is displayed in this third and final column. And as you can see, even in severe chronic illnesses like rheumatoid arthritis, osteoarthritis, you get a normal activation of pain inhibitory action in response to exercise. That's why it's more fun for those patients to do exercise than it is for patients with chronic fatigue syndrome or fibromyalgia. As you can see, fibromyalgia patient presents the same overlapping dysfunction. So it is a rather specific thing. But when you were watching this picture, you were not thinking of what is happening in the brain of this poor guy, but you were saying, yeah, but the bull and the stressful event of the angry bull chasing him is the thing that makes him distract from the possible nociception arriving from his skin. And of course that's true, and that's one of the reasons why we should actually control for emotional stress in studies looking into exercise-induced changes in patients with chronic fatigue syndrome. And that's possibly one of the major flaws of previous studies in the field, and we try to account for it in our current work. So we know that severe stress is not really able to activate this real powerful top-down pain inhibitory action, because in response to severe stress you get a decrease in crucial brain neurotransmitters like serotonin, like gamma aminobutyric acid, that is, and less availability of these powerful inhibitory neurotransmitters implies that there is less capacity of the brain to inhibit hypersensitivity, and you get more excitability of the central nervous system. Again, this is in line with what we see in patients with chronic fatigue syndrome, that they end up having this disorder in response to long periods of severe stress. So we studied the response of patients with chronic fatigue syndrome to exercise, and we nicely controlled it for the possible bias originated by emotional stress, by having all subjects, including healthy control subjects like this one, to also do a well-validated emotional stress test by watching horrible pictures on a computer screen, which actually triggers a lot of stress. And then we measured several things, including, of course, pain thresholds, but also cerebral blood flow. As you can see over here, we also measured, that's what you see, transcranial Doppler for measuring cerebral blood flow. And this is the output from the cerebral blood flow measurements. And here you see the autonomic measurements, but we haven't analyzed the autonomic measurements yet. We're still working on that one. But we have analyzed the cerebral blood flow data. So all participants, patients and controls, they performed both exercise and emotional stress tests. We looked at pain thresholds before and after. It was nicely randomized, but unfortunately there was no such thing as a dysfunctional cerebral blood flow. So we hypothesized in advance that cerebral blood flow would be impaired in response to exercise versus emotional stress, but the response in patients and controls was really similar. You might think that either one of them is patients or controls. It's not, it's just the left side and the right side. And these are the different conditions. Here you see, let me have a look, baseline, at rest, exercise response, and then the emotional stress test. So you can see that there is a normal increase in cerebral blood flow in response to exercise in patients with chronic fatigue syndrome as compared to healthy control. So unfortunately, this is not one of the pieces of the puzzle. So we know that there is a role for explaining post-exertional malaise in the immune system, the exaggerated immune response to exercise. We know that exercise winds up the sensitivity of the central nervous system. We know that the autonomic nervous system plays a key role in explaining post-exertional malaise, but we know that cerebral blood flow is unlikely to explain part of the symptoms seen in response to exercise in patients with chronic fatigue syndrome. But is it purely a biological thing, or is it also a psychological thing? We wondered whether expectations might account, at least in part, for the symptom increase following exercise. Because we know when it comes to fatigue or pain that it's an output product of the brain. Both fatigue and pain are typically regarded as input products to the brain, but in fact they are output products of the brain. So it always comes down to processing the incoming stimuli from the body and then working it out in the brain, and then you cannot ignore all these factors which also account for how the brain processes the incoming stimuli from your body. So therefore, anticipated consequences are a crucial part of possibly explaining how patients experience fatigue and pain increases following exercise. And this is also what we found and what people found in the Netherlands, that anticipated symptoms following even daily physical activities like stair climbing are actually related to the way they're actually able to perform the exercise, and also probably to the amount of symptom increases following exercise. So yes, symptom expectations do affect the way that they're able to perform physical activity and how they will experience the response of their body following exercise. And this is not the stair that we used for doing this experiment, because then it would have been a really expensive experiment, because this is one of the stairs in the atomium in Brussels, connecting the different balls of the atomium in Brussels, if you're familiar with the atomium. So the puzzle is nearly complete, at least for all that I have to tell you today, because I'm quite sure that there will be another piece of the puzzle which has not been addressed in this lecture or has not been studied up to now, but at least we now know that there is an important role for the accelerated immune response to exercise to explain post-exertional malaise in patients with chronic fatigue syndrome. We know there is a role for the patient's expectations to have increases in pain and fatigue following exercise. We know that the central nervous system is wound up and gets more wound up when it's responding to vigorous types of exercise. We know that the autonomic nervous system is responding poorly, especially the parasympathetic branch of the nervous system is not responding appropriately in the recovery phase following exercise, possibly interconnected with the immune system, so we get more understanding about how they respond so poorly to exercise. I really need to thank those funding agencies, those two charities. The MA Association has funded the study that I mentioned about the parasympathetic recovery or the lack of parasympathetic recovery following exercise. So a big thank you to the ME Association. I especially want to thank ME Research UK, because if it wasn't for them, we will probably no longer be able to study chronic fatigue syndrome in Belgium. It's much more easier for us to get funding for studying pain in cancer survivors, for instance, or pain in low back pain than it is for chronic fatigue syndrome. So a big thank to ME Research UK for supporting us all these years. This is the Pain in Motion group. As you can see, we try to have a lot of fun together. A big thank to all of them for this part of the work, especially a big thanks to Jessica van Oosterwijk and Mira Meus. And if you want to check us out on the internet, we're available also in the social media. Thank you.
Now we're scheduled to have questions at the end. Do you want to? Have we got any questions for Jo at this point?
In terms of the patient expectations, from personal experience, when one improves somewhat, one's expectations of what one can do just follow it back up. And then if you decrease, you're disappointed and your expectations are lowered again. How does that fit in with what we found so far?
Yeah, I know what you're saying, and that's a very tricky one, especially in terms of rehabilitation, of coaching patients through their grading phase of the rehabilitation program or exercise program. So yeah, that's a very tricky one, but I think it comes down on how you interpret as a patient, how you interpret the symptom increases following exercise. And it also comes down to closely monitoring and closely supervising the grading phase of any kind of physical activity or exercise program, that you don't do too much and that you make sure that you don't get too enthusiastic, which might trigger again a more pronounced immune response, for instance, which will make sure that you will get more symptoms. So yeah, I see where you're going, and I think that's a very important thing to highlight, especially for clinicians and patients. Yeah.
So if you're doing that exercise, presenting that model that the increased exercise results in that immune reaction, quite possibly would be fairly significant for patients, because it gives them a specific something to stop for.
To stop for, yes, but also I emphasized the fact that we see in many other chronic illnesses that you can also train the immune response to exercise. So that should also give patients hope that they can change the way their immune system is responding to their the amount of physical activity or exercise that they expose their body to. And normally we should also be able, and this has been shown also in many other illnesses, that we should be able to train the autonomic nervous system, for instance. There is also a lot of hope, of course.
Now, can I ask you something? The majority of ME patients after exercise experience pain in the muscles that they have used. This work that you've done looks at the central nervous system and the immune system response. Now, do you tie this in with the way of recovery in the actual muscle fibres? I know there's other research being done with that, but the two things should be tied together.
Yeah.
Because there is an abnormality in the muscle fibre and in the metabolism.
Yeah, thank you for that. I think that's also important to highlight, and I couldn't agree more that the two things should tie together, and I think we should also look into the interconnections, and I'm very impressed by the work done, including the work done by Julia's group on the local muscle dysfunctions, and I'm keen to learn more about that. But I don't think it's correct to say that when you feel the pain increases following exercise in the muscles, that this provides evidence that it is in the muscles, because that's a specific thing that we now understand from pain and from the increased sensitivity of the central nervous system, that you feel the pain wherever in your body, but it is actually primarily related to the increased sensitivity of the central nervous system. And I'm not saying that it is not located in the muscles, not at all. I think there is becoming more and more studies out there available that show that there are some abnormalities, at least in a proportion of patients with chronic fatigue syndrome, in the muscles. But what I'm saying is that that should not make us ignore the fact that there is an increased sensitivity in the central nervous system. So it's the both of them.
And I think if I can chip in as well, I think what would be the definitive study is to actually be listening to, I think it was Mark yesterday from St George's, would be to actually do muscle biopsies in those patients as they exercise, and so that actually the two things are measured at the same time, and actually being really joined up about and maximising the outputs from the studies, and being able to measure simultaneously in the same people what's happening in the muscle and what's happening in the brain. Norman.
Yes, that's an amazing set of experiments. But carrying on from the last question, there is now evidence that there is at least one more pathway regarding metabolism, and that is the purine degradation one. We first noticed this in neutrophils while they're just wandering around in a test tube looking for a pathogen. So they're using just motility and chemotaxis because there were no pathogens there. In half of the patients, we found there was another anaerobic process apart from glycolysis. And this has been modeled very mathematically by Lengert and Brüssel in Darmstadt. And it carries on from the production of AMP by the ADK reaction in muscles or anywhere where a lot of ATP is being used. And the adenosine monophosphate is deaminized to produce inosine monophosphate, and that goes down, and you need to re-synthesize the adenosine nucleotides, and this can take several days. So this is fairly new, but there's mounting experimental evidence that that goes on. And you will get then metabolites in the muscles or in immune cells, which take a long time to get rid of, and this is very damaging.
Absolutely. I think it goes back to what the lady in front was saying. We need to do those experiments, and we need to know what's going on in both the brain and muscle simultaneously, so that we can begin to answer some of these questions. Is it peripheral, is it central, or is it a combination of the two? Which I think is probably the answer. But sat in front of you in a row is the AMPK lady, it's first author of the paper, and two PhD students, both of whom are working on muscle in Newcastle. So, you know, there are real opportunities for us to look at this in more detail, but we need the tissue. Any other questions? Five.
Can I ask you a slightly unrelated but also related question? Is that why do you think that inflammation occur after exercise? Is there a physiological benefit from having inflammation, and then why you trigger the vagus nerve to down-regulate inflammation?
Sorry, could you repeat the question?
So really, I just want to know, I mean, they're often a reason why you develop an inflammatory response after exercise. So do we know this, whether there's any physiological benefit? And also then the regulatory pathways.
Yeah, that's a good one. It is actually of physiological benefit to have some kind of inflammatory response towards exercise, because you always, when you just look at the muscles, for instance, you always have at least some kind of very small tissue damage in terms of working muscles, and it's not really damage in terms of something bad, but it's normal that the muscles, they respond and they change in response to doing exercise. And therefore, it's normal that the immune response actually is there to decrease and to get rid of the slight pieces of damage which have occurred. And this is also important for making sure that the muscles will be able to adapt to the exercise. But again, it's kind of a homeostatic response, and you see that it will decrease when you do this same type of physical activity or exercise all over again. And you experience that yourself if you are doing exercises or kind of sporting activity which you have done for ages. And the first time you do it, it hurts a lot locally in the muscles, but after a while you adapt and the immune response decreases to that same type of sporting activity. So yeah, I agree, it's an adaptive response.
He's an immunologist, so.
No, sorry. It just, so therefore, because you mentioned about the exercise in localized muscle group, is that muscle group that actually have the anti-inflammatory action? So is again that systemic versus local control, where does it come from? So if you exercise, obviously, as you say, when you practice signal exercise, initially the first time it hurts in that muscle group that you exercise, and then of course you must have triggered anti-inflammatory response, and over time you don't feel that. But again, it's localized. The muscle group are not, in general, all the muscles less painful.
I suppose it's a systemic effect. Or...
Yeah, but what we know from exercise immunology studies is that these immune responses were not really identified locally in the muscles, but they were identified in the blood in general. So it's not that I understand your viewpoint, but it's not that we have enough evidence to say that it's a real local effect. It's more a general effect, but because the majority of those studies which I relied on to say that there is an immune response to exercise were done by taking blood samples pre-post exercise.
So can I just follow up on that a little bit? And say, was I right in thinking that you said that acute exercise caused an inflammatory response, whereas a chronic exercise caused an anti-inflammatory response? I'm just thinking in terms of our evolution, we were probably a lot more active. It was on the African plains trying to get food and being prevented from being eaten, and maybe this is more a thing of modern lifestyle where we don't exercise as much. Or is that just me that doesn't exercise?
Well, yeah, I guess you're partly right, but I think it's important to slightly correct the meaning of anti-inflammatory in response to long-term exercise. It's not that you get an actually literally anti-inflammatory response to exercise, it's just that the inflammatory response to exercise, when you do it regularly, decreases. And therefore the balance between pro-inflammatory and anti-inflammatory changes and gets more normal, let's say, but it's not that you get a decrease in inflammation following long-term exercise. You do get a decrease in the inflammatory response to exercise in the long term when you do the same type of physical activity all over again. So it's not literally anti-inflammatory, but it's a decrease in the inflammatory response. Okay?
Great. Any more questions? Brilliant. If not, I will thank Joe one more time. And then I'll introduce myself. The great luxury of chairing yourself. I would put mine together. Brilliant. So you're being tortured by me for the next two talks. It was a huge relief when I looked around the audience just stood there to see that Andreas is here, who's the MRI expert who's been working on the project that was funded by the MRC. And I've actually got them the wrong way round in the programme. So we're going to do the second talk first. And it's really important that I acknowledge again, as I did yesterday, my thanks to the MRC for funding this large cohort study, but to the team who've worked on this project to get it to where it is today. If there's any difficult questions about MRI, then we'll have to defer to Andreas, because as Jose Montoya said yesterday, what this is all about is bringing people into the field who are experts in their area to study a condition that hasn't always had the opportunity to access state-of-the-art investigations. So, although I stand here today presenting a study on cardiac MRI, I am by no means the expert in this field, and you will see when we come to the next talk that I'm also by no means the expert in cardiac MIBG. So if there's any difficult questions, I will look that direction to Andreas. So, as you'll remember from yesterday, part of the series of investigations that we performed in the patients who participated in the study funded by the MRC were that we were performing cardiac MRI, and we were also performing plasma volume. And as I mentioned yesterday, one of the major strengths of this cohort is that we performed a lot of investigations in patients, which allows us to look with a whole systems approach at abnormalities and begin to tease out what may be the associations between different abnormalities. So, for those of you that are not familiar with the field in terms of cardiac abnormalities in chronic fatigue syndrome, there's actually quite a considerable number of studies suggesting that there are abnormalities of cardiac function and cardiac structure in patients with CFS. There have been studies performed in Japan using echocardiography and using chest x-rays, which have come up with this suggestion that chronic fatigue syndrome may be a small heart syndrome. So using both measurements from chest x-rays and echocardiography, they've suggested that the cardiac structure is smaller in CFS. And more recently, we've published 2 studies funded by ME Research UK, where we took individuals, 20 patients with CFS-ME and 20 age, sex and activity matched controls, and performed cardiac MRI, structural, bioenergetic and cardiac tagging, and suggested that there may be reduced cardiac volumes using an MR technology. So what we wanted to do in this study is take our findings that bit further, and rather than having a separate cardiac MR study, to be able to look at whether or not the findings of the previous study were confirmed, and then to see whether we could begin to tease out whether there were any other abnormalities that might be associated with the changes in cardiac function. The current literature has suggested that the cardiac abnormalities that are present are secondary to deconditioning. So the reasons why individuals with chronic fatigue syndrome have smaller hearts is because they are inactive. So this is one of the things we wanted to look at. So we set out to extend our previous cardiac MR studies into a second, larger cohort, and we wanted to confirm our previous findings that there were abnormalities of cardiac function, and we wanted to go on using the large resource that we have with the MRC funded study to explore potential mechanisms. So as you saw yesterday, all the participants, as well as having autonomic function performed, they had non-invasive cardiac function using cardiac impedance, using something called the task force monitor, and they also had red cell mass and plasma volume measurements performed. All the participants in the study were recruited through a clinical service, and they all fulfilled the FACUDA criteria for the purposes of this study. We measured fatigue using the fatigue impact scale, that's generally our measure of choice, and they all attended the dedicated MR facility in Newcastle, and this is a dedicated research facility, and they had cardiac MRI performed using a 3T Philips scanner. They then subsequently, within a short period of time, went on to have total red cell and plasma volume measured using a dilution technique, using 51 chromium labelling of red cells and iodine labelling of serum albumin, which I am led to believe is a very standardised way of measuring plasma volume and red cell volume. So, in terms of what we found, we were able to perform cardiac MR in 47 of the participants, who were then matched case by case for age and sex to 47 controls. And the mean length of the history of the CFS cohort was 14 years, but there was a huge range of length of history, which was something that we were very keen to do, so that it allowed us to explore whether the issue of deconditioning was a potential underlying problem. And we were able to confirm our previous findings that the CFS patient group had significantly reduced end systolic and diastolic volumes measured from the cardiac MR, and they had reduced end diastolic wall masses, which is just shown, the values are shown here on this slide. So that allowed us to confirm in a larger cohort our previous findings that there is a cardiac abnormality in this patient group. 41 of the CFS patients and 10 of the controls who were matched group-wise also had plasma volume and red cell mass measured, and these are the values that we were able to find in the CFS patient group. We were able to show that 55% of the CFS patient group had had values of red cell mass below 95% of the normal expected value, and 28% of plasma volume was reduced below the expected value. There were no significant differences group-wise between the CFS and the controls, but one of the problems, as Andreas pointed out yesterday, is that we only had 10 controls. And most importantly, there were very strong positive correlations between total volume and the cardiac abnormalities that we found. So you can see on the graphs here, these are showing plasma volume and end wall mass, wall papillary mass and wall mass, and there's a positive correlation between plasma volume and the end diastolic cardiac volumes. So these relationships held true for plasma volume and red cell volume. In addition, there was a significant negative correlation between fatigue severity and lower plasma volume. So the more fatigued patients were, the lower was their plasma volume, and there was quite a wide scatter, but it was significant, that correlation. And most importantly, from our perspective, was that there were no relationships between any of the MR or volume measurements and length of history, suggesting to us that it was unlikely that deconditioning would be a cause for the cardiac abnormalities that we were finding in this patient group. So in conclusion from this study, we've confirmed in a second larger cohort that there are cardiac abnormalities similar to the ones that we've seen in the previous published studies. So we extended our original study to include further assessments within the same individual and shown an association between cardiac volumes and plasma volume. There's no relationship between the length of the disease and these MR abnormalities and plasma volume, suggesting that the findings are not related to deconditioning. And we are hypothesising that these reduced cardiac volumes may constitute a vulnerability, a pre-existing cardiac abnormality, which may ultimately be a vulnerability for developing CFS, but clearly we need further longitudinal studies in order to support this hypothesis. Perhaps most importantly, in terms of the findings from this study, is that there was a relationship between plasma volume and fatigue severity, suggesting that the more fatigued patients are, the lower their plasma volume is, which potentially gives us a therapeutic target. So in terms of next steps, in Newcastle we have an MRC Confidence in Concept programme, and what we've now done is submit an application to that scheme in the hope that we'll be able to attract some funding that will will allow us to look at whether or not if we increase plasma volume by intravenous boluses of fluid, whether this impacts upon cardiac volumes. So if we are able to establish proof of concept, that may then allow us to progress to a full study to see whether intravenous boluses of fluid do influence plasma volume, and whether by influencing plasma volume does that lead to changes in cardiac volume. And again, thank you to the MRC for the opportunity to pull together this cohort. Shall I do the next one before anybody asks me any questions? Brilliant. Thank you. Fine. So the second study, and this is a small study. George Petridis is a consultant radiologist who has a special interest in MR technologies and nuclear medicine. So he is a great help in terms of analysing this data, and you will see as we go through, it's a very small study. The intention at the beginning of the MRC study was that everybody would have cardiac MIBG, but unfortunately in the interim between applying for the funding and securing the funding, the cost of 1,2,3 iodine benzyl guanidine, MIBG, went through the roof. So it increased tenfold and had to be imported from America. So we had to compromise with a fixed envelope of funding how many scans we could do. So as a result, we made a decision with this particular component of the MRC cohort study that what we wanted to do was to get some pilot data to inform a potential future larger study. So what I'm going to present is a small cohort of participants who had MIBG, but they are all participants within the larger cohort study. So, as you've heard quite a lot of in the last 24 hours, there are a large number of hemodynamic abnormalities documented in CFS, which suggests that there are abnormalities of the autonomic nervous system, which are and particularly in the aspect of the autonomic nervous system that regulates cardiovascular function. I've just told you about the cardiac abnormalities that have been detected, and the purpose of the MRC study originally was to try and delineate whether the autonomic abnormalities that we're seeing originated centrally in the brain, or whether they may entirely or in part originate from peripheral abnormalities of innervation of the cardiovascular system by the autonomic nervous system. As we've heard from Yo, there is evidence that there's sympathetic overactivity in CFS, and there's also evidence that there's impaired baroreceptor sensitivity in CFS patients, particularly in response to physiological stresses. Other studies have also confirmed reduced heart rate variability, particularly related to parasympathetic function and dysautonomia. But despite all those findings, it still remains elusive as to where the primary abnormality lies in the autonomic abnormalities that we can find so frequently in this patient group. And as a result, we don't have any therapeutic targets in terms of the autonomic nervous system. Whether or not we should be focusing on defining the abnormalities in central innervation of the autonomic nervous system, or whether or not it's a peripheral abnormality. We all have our theories, but we don't necessarily have the conclusion as to where the abnormalities lie. So what this particularly focused study aimed to do was to use a technique called MIBG imaging as a quantitative tool to look at cardiac sympathetic nervous system innervation very specifically. It's been used in a range of different conditions. It's used quite a lot in Parkinson's disease to look at autonomic dysfunction. Studies from Newcastle have looked in forms of dysautonomia and confirmed that in conditions such as carotid sinus hypersensitivity and vasovagal syncope, that abnormalities of cardiac sympathetic innervation occur. So we wanted to use that technique in this specific patient group. We wanted to explore whether or not there was an abnormality of cardiac sympathetic innervation, particularly in CFS, and to map these abnormalities to the cardiac MR and other measures that we were also taking in this cohort. So what is MIBG and what does it measure? Well, it's a form of SPECT scanning. It's a radionuclide imaging technique. It's very good at looking at cardiac sympathetic innervation. And radio-labelled MIBG is an established sympathetic imaging technique useful at looking systemically at organs richly innervated by the sympathetic nervous system. And that will make sense as we go through the next few slides when I get to the next steps as to what we're now beginning to do with the images that we've now obtained. And for cardiac SPECT, what it is able to do is that they are able to develop a region of interest around the heart and then compare that to mediastinal levels of MIBG and produce a ratio of heart to mediastinal MIBG that's detected. So this is quite a complicated diagram to show you, but MIBG is an analogue of guanfidine, and it's taken up by postganglionic presynaptic nerves, specifically here in the cardiac myocyte area. So the MIBG is injected and goes systemically, but specifically in terms of cardiac function, after depolarization, MIBG is released into the cleft, like noradrenaline is, but it's not metabolized. So as a result, it's able to be visualized. And the scanner is a bit like an MRI scanner, and the patient has an injection of MIBG, which is administered intravenously, and then imaging is performed at rest and after 10 to 30 minutes. The subsequent of the images are analysed where they compare regions of interest, the heart, compared to the mediastinum, to produce what's called an HM ratio. So just to reiterate the types of patients that are included in this, they're all Fukuda patients. They've all been to a specialist service. This patient group have had depression screened for using a DS, the skids, so they absolutely do not have a history of a major depressive episode. So we are considering this to be a CFS group without depressive comorbidity. We use a task force monitor, which is the autonomic testing system that we use in Newcastle, which allows us to derive a range of autonomic parameters, including heart rate and blood pressure variability, bioreflex sensitivity, using spectral analysis and the autoregressive model. And we quantify fatigue using the fatigue impact scale. So as I said, this is a small cohort, pilot study, the posters next door. There were 9 CFS patients included in the study, and we were not able to study any controls with this particular technique. So we have had some difficulties finding a control group to compare our patients to. So the mean age of the patients that were included in this was 51, 5 of them were female. The length of history of this particular group was 17 years, again with a wide range. Mean HM ratio was 2.94. Initially we thought this was Nijs suggesting that they had sympathetic hyper-innervation of their heart, but depending upon which particular cohort of controls you look at, this may or may not be considered to be on the high side. Importantly, there were no significant correlations between HM ratio and cardiac structure or function or length of history. But there were positive correlations between HM ratio, so higher HM ratio shows higher sympathetic innervation, between barrow reflex sensitivity and the balance between sympathetic and parasympathetic autonomic function in terms of the variability of diastolic blood pressure. Remember, we showed in cardiac MRI that there were changes in end diastolic volume, and what we were able to show here was that there was a shift in the balance between sympathetic and parasympathetic function. There was a significant positive relationship between high-frequency heart rate variability. This is considered to be predominantly a measure of parasympathetic influence upon heart rate variability. So lower high-frequency heart rate variability associated with lower HM values. And again, there was a shift in the balance between parasympathetic and sympathetic influence over the system in terms of heart rate. And the final finding was that there appeared to be a relationship, again, very small numbers of patients, but it was significant, was that when we measured, compared the HM ratio against fatigue severity, it appeared that the more fatigued patients were, the lower was their HM ratio, and so the lower was their cardiac sympathetic innervation. So very preliminary data. I've absolutely acknowledged that this is a pilot study, but what we are beginning to see is that impaired cardiac sympathetic innervation measured using MIBG uptake appears to be associated with increased fatigue severity in CFS patients, which suggests that it may be a primary cardiac abnormality that underpins the autonomic abnormalities that we detect so frequently in patients with CFS. So certainly something that we think is worth further exploration. In terms of next steps, it's always very important to sort of emphasise where this is going next. And one of the things that George, while looking at the scans, detected was that there appears to be in patients, and as I said at the beginning, MIBG goes everywhere in patients when the scans are performed, was that he was seeing some quite unusual patterns of how MIBG accumulated in the liver in the CFS patients and the amount of MIBG that there was in the lungs of patients. He found that, considering that he looks at MIBG scans a lot of the time, that the patterns he was seeing in this cohort were different than he had expected. So we're now also producing ratios of, for lung and liver measurements to see whether or not actually, when we look at how MIBG and sympathetic innervation is happening out with the heart, whether or not there may be abnormalities in a more systemic way, rather than just focusing on the conventional use of the tool, which clinically at the moment it's used conventionally to measure cardiac innervation. So at the moment we're doing further analysis measuring liver and lung MIBG levels. Cool. Any questions? Oh, please don't ask any any difficult questions, because I might not be able to answer them and then I'll look stupid. Oh, Stephen's got a question at the back. That's going to be really hard, isn't it?
No, it's not. I've been listening and making extensive notes.
Oh dear.
So I think what this really illustrates, and you said it sort of at the beginning, I think Joe may have said it too, how important it is to do this joined upness and studying the integrated effects of chronic fatigue syndrome on the human physiological system. And the well-worked up patient, which is what you're describing, is a fantastical way of discovering, how different organisms interact with each other. And I just wonder, as I hear this, whether if we were going to put an inventory up about what measurements we should be making on these patients, we've heard several examples this morning, different ones, to try and capture the complex interrelationship between blood levels and autonomic nervous system, blood volume, which you were talking about, and the central nervous system that Joe was talking about. So, do you think that would be something quite useful to do? Because obviously, clinically, we need to have diagnostic tools to help us definitively diagnose some of these patients, so that we can convince our health services that this is something really, really important and real, without us just solely relying on the symptoms.
Yeah, and actually, one of my colleagues who's here from Poland, Pavel, has done a very similar thing. He's been round visiting people, got lots of ideas, and gone back to Poland, where at the time CFS wasn't recognised as a disease, and has come up with a project and invited people to participate. And the project includes most of the components of what we do, plus other things that he's interested in and picked up along the way. And you've got 100 people in each arm of yours, half of whom are having graded exercise therapy, aren't they? But there's a huge amount going on that I think you're absolutely right. I think it's really important that we all capitalise upon what each other is doing and learn from each other, and make sure that we do studies that re-study the same things so that we can get confirmatory evidence from other centres. That's really important, so that we get away from this single observations in single centres that never go anywhere. And to me, if there's any bit of this study that anybody's interested in, people would be more than welcome. What we need is pairs of hands to do the analysis. That's the sort of rate limiting step at the moment. And if people want to get involved, then and have data, then do let us know. But going forward, I think you're absolutely right. We need some consistency about how we do measure things, so we're all measuring the same thing. And then as a result, I think the amount that the steps we will take towards getting the answer will be much bigger. Cool. Charles?
In relation to the first study you were talking about with the cardiac volumes, were these patients otherwise cardiac normal? Their cardiac function, as far as you know, was normal. They didn't have any abnormal symptoms that were hypertensive?
No. Well, no. So, actually, surprisingly, few of them had abnormalities of autonomic function. So, when we've compared autonomic function, and the slide yesterday, which I rattled through, surprisingly, most of them didn't have the abnormalities that we have seen in previous studies, which we're finding quite a lot in this cohort, with various different neuropsychometric measurements, symptoms, which makes us think that some of the previous observations from perhaps our own and other studies may be related to the co-founder of depression. So this cohort being clean from depression means that we believe that we're studying CFS in its very purest sense. And it may be that some of the autonomic abnormalities that we found in the past may be related to the presence of depression. And we're comparing our previous ME Research UK funded cohort to the patients who participated in this study to look at some of that.
So these cardiac abnormalities weren't predisposing people to failing says
No.
algorithm.
No. So there was no, on structural cardiac MR, there was none of them had any structural abnormality.
Yeah, I actually have two questions. First, I'm not very familiar with this MIBG method. It's something to measure the sympathetic innervation, but is it qualitative or quantitative?
It's quantitative.
So it tells nothing about the strength of signals coming down from the brain?
No, that's absolutely correct. So it's very localised to what is the sympathetic innervation of the heart. That's specifically what it's measuring. So it's not looking at innervation from the brain, it's looking at the cardiac innervation.
Okay. And the second thing is, how do you actually interpret the those end diastolic and systolic volumes? Because I was thinking lower red cell volume, lower plasma volume, Frank-Starling mechanism takes over.
So the end diastolic, I'm looking at Andreas now, the end diastolic volumes and the end systolic volumes are literally measured from the scans. So Tim Hodgson, who's one of the radiographers, he physically has measured the volumes. Yeah, Andreas is nodding. So they're an actual measurement of cardiac volume during function.
That was not my question, is that how do you interpret this in terms of all the other findings? I mean, how does it take place? Why does it take place? Because I was thinking maybe it's secondary after the conditioning, losing a lot of fluid, lower blood volume. So this would be the finding I would expect in a dehydrated person due to Frank-Starling mechanism.
Yeah, absolutely. So that may well be part of the pathophysiology. It may be that a lot. Anecdotally, in the literature, there are descriptions of patients benefiting from intravenous boluses of fluid. In America, people get admitted to hospital to have intravenous boluses of fluid, and there's anecdotal reports of benefit to them as individuals.
Yes, I have administered some myself.
Yeah. So it may well be that although we didn't find that there was a significant difference in plasma volume and total volume between patients and controls, I acknowledge that there were only 10 controls in the plasma volume study. But when we looked at the percentage you had values below 95% of the expected, there were actually quite a chunk of the total cohort who had volumes which might be considered to be low. So to me, the next step is to actually perform some. This is an observational study. What we need to do, as people said yesterday, is we need to start doing some intervention type studies to see whether we make any difference to symptoms and cardiac function as a result of intervention. Now in my clinic, what I do is I always encourage people to drink 2.5 litres of water a day. And that often will make people feel dramatically better. And if we believe that these patients have low blood pressure, which I do, and that they have a tendency to drop their blood pressure, something that we find very frequently in people with fatigue-associated diseases, it may be that it is a volume problem, at least in part, and that by increasing the blood pressure, the head of steam that's perfusing all our organs, etc., by either oral means or intravenous means, that might ultimately improve people's symptoms. But, that's the study to do.
Hi, Julie. Two questions, if I may. You've previously shown that there are abnormalities in cardiac muscle energetics. So the first question relates to whether you see any association between reduced plasma volume and cardiac muscle energetics, because my thinking is that if you've got a smaller heart, is that having to work harder to create, albeit a reduced stroke volume, but...
Yeah, so we didn't perform cardiac bioenergetics in this new cohort, and equally we didn't perform muscle bioenergetics. That wasn't part of the funding for this particular study. So in terms of the muscle function, peripheral muscle function, and how it responds to exercise, we don't have that information in this cohort. and we don't have the cardiac bioenergetics, but it may well be that because you have a smaller heart, you're not generating as greater cardiac output, and as a consequence that perfusion is less, so peripheral muscles, etc.
And just quickly, the second point, given what we've heard about inflammation, have you had a chance to look to see whether there's any association between inflammatory markers and your cardiac abnormalities?
So the big luxury of this cohort, which will be hopefully next year's presentation, is that we have had a guy who's done an MPhil, James Clark, who's gone on more recently to go to medical school, and he's got a brain that works in a different way than me, and he has been looking in a whole systems way, modelling with the team of biological modellers down in Newcastle University, looking at all the parameters that we've got, specifically looking at immunological with Phi's input, the HPA axis with Stuart Watson's input, and the autonomic parameters. I can't remember exactly the measures that have come out now, but he's writing a paper as we speak, taking the whole cohort with all the parameters, taking a systems approach to it, and there are things that interact from an immunological point of view, an inflammation perspective, with the autonomic parameters. So that's one of the papers that's in preparation at the minute. Can you remember the answer, Stuart? But no, it's very, very complicated.
Hello, just in relation to the first presentation, and you're considering the possibility of deconditioning versus a primary cardiac abnormality or malformation. Did you take a history of the patients, get some information on their activity levels prior to illness?
No. So their activity levels were measured using the IPAQ. So we have their activity levels at the time they were studied. We know the length of their history, but we don't know what their comorbid activity levels were. What are you thinking?
I was just wondering, you could do some sort of ranking test. So if some of them were very active, prior to illness, you could then see if, how they then ranked subsequent, if you see what I mean. There would be a way statistically, perhaps, looking at, again, it's self-reported, so it's not something you can take at the time. It's much more difficult for people to reflect on what they did in the past. But you could just see if the people who had worse cardiac performance had a lower activity prior to illness, or whether even the people who were very active prior to illness still have if there wasn't a relationship, if they still have very low cardiac outputs subsequent to illness.
Stuart, this is really bad when people who you actually work with are asking you the difficult questions.
An easy question.
Oh, good.
Unless people are dehydrated, then the Holter fluid is just going to get weed out, isn't it?
Well, or it may be that they're not dehydrated. It may be that they're putting volume in the wrong place.
Okay. Then what about EPO?
Somebody asked a question about EPO yesterday via Twitter, the red cell volume wasn't significantly different between the patients and controls. EPO would be a more aggressive intervention to trial. So for me, the first step would be to look at volume. But people describe on the internet benefit from EPO. But I've never seen, to my knowledge, I've never seen a trial of any sort. It's all anecdote. Norman, is it back?
I've got one from
Twitter.
I remember years ago a letter in either the AFME magazine or the ME magazine by a woman who had occasional blood transfusions for some other illness, but she reported that when she had a blood transfusion, it improved her ME symptoms. So certainly blood volume is an important thing. I've got many PowerPoint slides from Dr. Paul Cheney, who gave numerous talks in the United States. He did a lot of work on diastolic relaxation times. Unfortunately, he never published anything, so it's too bad it can't be referred to. But he did think that mitochondrial dysfunction was involved, because after all, heart muscle is about 30% mitochondria.
I agree. Any other questions?
Yeah, Julia, I've got one from Twitter. Jack is asking, is there any sense of time? If things stay at this pace, how long before any test studies reach mainstream? Decades? Question mark.
No, so we have the confidence in concept. So you have to prove something works. So science is very frustrating. Nothing happens as quickly as anybody would like. And I spend my life writing unsuccessful grants. But occasionally one is successful, and that's really when the excitement starts. So what we now have to do is take the data that we have and begin to look at where the opportunities lie. And certainly there are opportunities around the observations of plasma volume because of the anecdotal reports that there are in the literature. And if we can get proof of concept, and the application for that has been submitted, so we will hear whether the scheme has been refunded by the MRC in Newcastle in the next few weeks. So that, if we are successful, should happen within the year. And then if we have that, then we need a proper study. So if we have proof of concept, then we would be looking for an EME. So decade is a long time, but probably realistic in science. All the scientists are all nodding sagely in the audience. Yeah. Oh, cool.
So you measure cardiac volume, and did you do cardiac function as well?
So we measured cardiac volume, and we did cardiac tagging. We're still doing the analysis of the cardiac tagging data at the moment. When we look back, we wish we'd done cardiac bioenergetics again, but we didn't, because that involved slightly longer for the patient in the scanner. And these scans they're quite hard for people to participate with because they have to lie on the front, which is quite hard for people. We went for what gave us the quickest wins, and in retrospect, we wish we'd done bioenergetic function as well.
Yeah. They have to lie on their front for the cardiac scans.
Andreas?
Okay.
Not for the ones we did.
Oh, not for these ones, sorry. It's the bioenergetics, they have to lie on the front.
Okay. I don't think that if we did bioenergetics that we would require that, but I'm not my cardiologist.
Yeah, which ones? Because in the previous study we led them on the front, because I was a control. Anyway, sorry.
Yeah, I can imagine how that can be.
Yeah.
Yeah. So, did you measure your cardiac volumes manually?
Yes.
This is the man that did the work.
we'll talk later, Andreas, but we can talk about how to do it semi-automatically and the cardio dynamics not on the front.
Yeah, well, I think Andreas is suggesting that they didn't, but in the previous study I did lie on my front, and I remember it well. Yeah, the gentleman at the back.
I'm just wondering if there's anything useful to see here about comparisons with other patient groups, particularly the long liver results.
Yeah. So it is very interesting, because one of the things that we also did was liver MR and measuring liver volumes. And Andreas and a cohort of medical students spent many hours in a darkened room drawing round images of livers to try and get liver volumes, and it's been really difficult to get a consistent methodology, because what we were asking people to do was do a Valsalva. So we were trying to take pictures of the liver during the phases of the Valsalva to see what happened to liver volume. And we can see in individual scans that as the systemic blood pressure drops, the liver squeezes, and we know from animal work that the liver and the spleen are the major capacitance vessels for homeostasis, for blood pressure homeostasis. So what we really wanted to see was what contribution in humans does the liver make to blood pressure regulation. And that's been extremely difficult to quantify in a way that we're happy is consistent and reliable. And Andres is shaking his head in frustration. So the liver is really interesting because you can watch it shift in huge, what to me on the images looks huge volumes during a Valsalva, and one would imagine that that sympathetic innervation of your liver capsule, which we know from studies in animals is happening during the Valsalva to keep your blood pressure perfusing your brain, that if we could quantify that in some way, that would be really interesting to then go on and explore what happens when you have a liver transplant, what happens in autoimmune liver disease where primary biliary cirrhosis, which is where I started in all of this, is a fatigue-associated disease. We know liver transplant patients are profoundly fatigued after their liver transplant. So if we can get the methodology right, actually being able to look at what the impact of an autonomic stressor like a Valsalva has upon liver response to changes in blood pressure would be really useful. And it's just the MIBG is sort of almost teasing. And I think probably when I sort of mentioned to Stuart about blood being put in strange places, your liver's like a big sponge. And at the minute, we don't know what contribution in humans your liver makes to blood pressure regulation. We know in animals, if you're having hemorrhagic shock, it's your liver and your spleen that come to your rescue. But we don't know in humans what role it plays in blood pressure regulation. And that, I still have some minor optimism. We may get something from the liver MR scans at the minute, but I think Andreas may have lost the will to live with it by now.
I might be able to give Andreas some will back.
Oh, good. Well, George came up with some type of automated measure of volume. So, but in the end, I don't think even that. But yeah, that would be brilliant.
Because with brain MR imaging, there's been loads of groups across the world, America, the UK, Europe, that have developed these analysis methods, automated methods, quantifying, robust, reliable, validated methods. But for other parts of the body, the quantification, like calculating cardiac volume, looking at liver, and being confident that you've segmented out, that's all of the liver you've got, and Nijs mentioned this. There hasn't been that.
Yeah.
What's been done for brain imaging. And there's a group of us in Bristol, image processing people, who are experts in image processing of stars in the sky, or they process other images.
You two need to talk. That would be marvellous. Thank you. Brilliant. Yeah. You can have a date at the bar now. On me, I'll buy the drinks. Brilliant. Well, thank you very much, everybody. We're rendezvousing again at 11. So coffee's out.